we're going to go ahead and describe 3 different applications that we're going to be seeing in this class. So the first one is gene expression. That was the first use of microarrays, was to measure gene expression. And in this cartoon, I'm showing you how the Affymetrix gene chip array works. What they do is for every gene, which is represented in this cartoon with the long line at the top. So this is the transcript of the gene, which we know its sequence. So what Affymetrix does is it takes 11 locations on this transcript, and it creates a probe that matches each one of these locations. Right, so you can see the 11 blue little lines underneath the transcript. Those are the probes, and we're showing in the cartoon where on the transcript they're going to match. Now, one other little detail about this is that in this array, the probes are towards the 3' end of the transcript, towards one of the 2 ends. That's because of the way RNA degrades. It tends to degrade more from one side to the other. So this company, for this particular product, they target the side that degrades less. So now you have, for each transcript, you have 11 probes, and they're scattered around the array to avoid confounding a location on the array with gene. You label RNA, you put it on the array, and if this particular transcript is abundant, there'll be many copies of it, and you'll get a lot of hybridization. So there'll be a lot of label on each one of the 11 probes. So you can see them here. I'm showing you in this cartoon one specific probe that is represented by a little square, and you can see that it's pretty bright. And we can look for consistency as well. Are all the other 11 right? And if they are, then we're going to assign a large number to this particular gene, which is supposed to tell us that this gene is highly expressed in this particular sample. So that's how microarrays can be used to measure gene expression. Now notice I've just showed you one gene, but we do this for all 20,000 of them. So for each of the 20,000 genes, we select 11 probes, we put them on the array, and we get data from which we can quantify gene expression for each gene. So another application of microarrays is genotyping. So in the first lecture, we talked about single nucleotide polymorphisms and how different humans can have different alleles. You can either have 2 of the same or 1 of each. And there's 2 ways. There's 2 different ones. So you can have 2 of, in this particular cartoon, you can have 2 A's or 2 G's, or you can have an A and a G. So if we want to measure this, if we want to know which of these 3 possibilities this particular human has, we can use microarrays, and we can do it for many SNPs, single nucleotide polymorphisms, at the same time. And in this cartoon, we're showing you just one SNP, and we've designed 2 probes. One probe hybridizes to the version of the gene of the piece of sequence that has the A. The other probe is designed to hybridize to the piece of sequence that has a G. So if you're an AA, if you have that genotype, then you will only get hybridization on the probe for allele 1. So that probe will be much higher than the second one, and that will tell you that it's an AA. If you're an AG, you'll have both, so they'll be about the same, and that'll tell you it's an AG. And if you're a G, you'll have the second allele will have a large intensity, while the first one will not, and that'll tell you it's a GG. So remember, and because this is a microarray, we can have millions of probes, and we can genotype millions of SNPs at the same time. That's a very useful thing to be able to do. These arrays are very popular for what are called genome-wide association studies, where they genotype many individuals, some of them with a disease, others without it, and they try to decipher which locations on the genome are associated with differences on the genome associated with disease of interest. And the third application we're covering today is the detection of transcription factor binding sites. And during the first week, we talked about how the genome is more than just sequence. There's all these chemical events that occur around the genome. And we want to be able to measure this in some way. So one example of something we might want to do is we might want to know where on the genome a specific protein is bound. A very common example are transcription factor, which are proteins that start gene expression. So we have DNA, and we want to know where is a specific protein bound. In this particular cartoon, the protein is symbolized with a little red diamond. So you can see that there's some pieces of the genome have it, others don't. So you start by fragmenting the DNA. There's techniques for doing that. So now you break it into little pieces. Some pieces have the protein, others don't. And there's another molecular biology technique that lets you divide the genome into pieces that have the protein bound and pieces that don't. So now you take the DNA the part of the DNA that did have the protein, you hybridize that to what is called a tiling array. This is an array that has a probe for all locations on the genome, or a selection of locations on the genome. If the probe lights up, that means that the location associated with that probe is a location where this protein was bound. Now the cartoon's a little bit more complicated than that. We have on the top, we have another microarray. That's because due to measurement error that we will describe later in the course, it's not that reliable. These intensities aren't as reliable for different reasons. So what one of the things they do to control for this is they also hybridize the total DNA as a control, and then they compare. Instead of just going by the intensity of a specific probe, they compare it to that same probe on the control array. And this is used to measure protein binding, such as transcription factor. So we've learned 3 applications of microarrays. There are others. In this class, we're going to see the data produced from these techniques, and knowing how they were produced actually helps us make decisions about how we analyze the data.